New York City is a pretty busy place. The traffic, the people, the messaging, the systems of transportation. But when comparing a living cell to this great city, New York pales in comparison. A cell is so complex that scientists have only scratched the surface of what really goes on in there. However, even with our limited understanding, what we do know is that the inner workings of a simple cell are simply extraordinary.
It's almost like there's a miniature city in every cell, and everybody has their own little job. If all the traffic lights decided to all just go crazy, you have issues that not only do we fix the light, but also we have traffic issues, we have crime issues, we have problems that snowball from one part breaking down.
Amino acids are the most basic building blocks of life. Each amino acid is an organic molecule that has a carboxylic acid and amine group attached to the same carbon atom. Here are two organic molecules that have the same composition but are mirror images of each other. These are known as right-handed and left-handed amino acids. Amazingly, living organisms only use and produce left-handed amino acids. Scientists have no explanations for this. When amino acids are created in a laboratory, an equal number of left-handed and right-handed amino acids are produced. It's a fascinating mystery that living systems only produce and use left-handed amino acids. Equally fascinating, these amino acids have many different functions. One particularly important function is its role as the building blocks of proteins. Proteins are linear chains of amino acids. Every protein is chemically defined by its unique sequence of amino acid residues. Just as the letters of the alphabet can be combined to form a variety of words, amino acids can be linked together in varying sequences to form a vast variety of proteins. Even the simplest life generally has thousands of proteins. These proteins are essential to organisms and are involved in virtually every process within a living cell. Proteins have structural and mechanical functions. For instance, in muscles and the cytoskeleton, they help to maintain cell shape, while other proteins are important in cell signaling, immune responses, cell adhesion, and the cellular life cycle. One particular protein, called a motor protein, transports various cellular cargo, like energy-producing mitochondria, to cellular neighborhoods in need of fuel. They can also provide the pulling power needed to separate chromosomes during cell division. Amino acids carry information from one part of a cell to another, as well as to other cells within the organism. So as you can see, without amino acids and proteins, basic cell structure as we know it could not exist. Other important types of proteins are enzymes. Enzymes are catalytic proteins that have special slots which hold other molecules to make chemical reactions possible. There are over 2,000 enzymes, and each one is used to enable a chemical reaction without ultimately being altered itself. It's been observed that the slowest biological reaction would take 1 trillion years without an enzyme, but the same reaction takes only 1/100th of a second when the enzyme is present. Pretty impressive. Adenosine triphosphate, otherwise referred to as ATP, is the energy source the enzyme uses to enable the reaction. Living organisms require and manufacture these enzymes along with all the other proteins. Let's briefly examine how proteins are created.
This animation demonstrates how the digital information encoded within DNA is used to direct protein synthesis. This is a DNA double helix containing the digital code which directs the cell in all aspects of operation. And here we see a protein complex called an RNA polymerase traveling down the DNA strand. As it moves down the strand, it carefully unwinds the DNA, preparing it for transcription. Inside the polymerase, we see a single-stranded copy of the original instructions being assembled as individual bases are positioned and added to the growing strand. A stop code marks the end of the protein specification, at which point this copy, known as a messenger RNA transcript, exits the polymerase and heads towards a two-part chemical manufacturing machine called the ribosome. While the messenger RNA moves towards the ribosome, transfer RNA molecules attach to specific amino acids in preparation for assembly. As the messenger RNA transcript passes through the ribosome, the process of translation begins. Using the instructions encoded on the messenger RNA as a template, the transfer RNA molecules align specific sequences of bases to corresponding amino acids, creating a protein chain. As this chain exits the ribosome, it is met by chaperones which prevent premature folding, while escorting the protein to a barrel-shaped machine called a chaperonin. This machine helps fold the protein into the precise shape required to perform its function. Although it is unclear how the chaperonin achieves this, we do know that accurate folding is essential in order for the protein to accomplish its intended function. Once the protein is complete, it is released into the cytoplasm to do its job.
As you can see, protein synthesis is an amazing process which occurs constantly in our bodies. Now, get this. Ribosomes are made from RNA and proteins, so the ribosomes are made of the very proteins they manufacture. In fact, it takes over 150 existing proteins to manufacture just one protein. So in essence, it takes a protein to make a protein. It's this chicken and egg scenario that continues to baffle scientists. Were discovered over 300 years ago, it was thought that the structure of a cell was very simple. With the inventions of the electron and proton microscopes, we now have a much clearer understanding of the intricacies of life. The cell is an extremely efficient and well-networked computer system with thousands or even millions of processors and billions of bits of information. Cells are the structural and functional units of all living organisms. Some organisms, such as bacteria, are unicellular, consisting of a single cell. Other organisms, like humans, are multicellular, having many cells. In fact, it's estimated that an adult human has over 100 trillion cells, and each cell is an amazing world of its own. It can take in nutrients, convert the nutrients into energy, carry out specific functions, and reproduce itself as necessary. Even more amazing is that each cell stores its own set of instructions for carrying out each of these activities. Let's take a closer look at how a cell works.
The first thing we see is the cell membrane. Much like the firewall software on your computer, the membrane contains protein gatekeepers, allowing only those components into the cell that belong and rejects all other components. Once we pass through this membrane, we see the organelles, or the organs of the cell. Ribosomes are the computer-controlled protein manufacturers of the cell. By fastening to an mRNA and using it as a template, the ribosome arranges amino acids in the correct sequence to form a particular protein. This process is known as translation. Golgi body. Next, we find a Golgi apparatus, also known as a Golgi body. Found in both plant and animal cells, Golgi bodies package and store proteins. Much like a post office, the Golgi packages and labels items, which it then sends to different parts of the cell. It's composed of membrane-bound stacks known as cisternae. Each cisterna is made up of a flattened membrane disc, and its primary job is to modify proteins, but it's also involved in the transport of lipids around the cell and the creation of lysosomes. Lysosomes. Lysosomes are spherical organelles which dispose of all the waste in the cell. These tiny organelles contain acid hydrolase enzymes that break up waste materials and cellular debris. The membrane around the lysosome allows the digestive enzymes to work at the 4.5 pH they require to break down the debris and dispose of it. Without the lysosomes, the cell would become overwhelmed with debris and ultimately self-destruct. Mitochondria. Mitochondria are the power plants of the cell, generating most of the cellular energy known as adenosine triphosphate, or ATP. In addition to supplying ATP, mitochondria are involved in a range of other processes, such as signaling, cellular differentiation, cell death, the control of the cell cycle, and cell growth. Nucleus. The nucleus is the control center of a cell. This is where we find most of the DNA which contains the instructions needed to create other elements of the cells. Like the motherboard of a computer controlling its components, the nucleus directs the growth, metabolism, and reproduction of the cell, among other things.
Life requires fully functional DNA, RNA, ATP, enzymes, and other proteins. If any of those components were missing or not working properly, life would fail to exist. This continues to generate several unanswered questions. How could non-living material develop the hardware and software known to be required by all living organisms? How did nature develop the arbitrary protocols for communication and coordination among the thousands of computers in each cell? These kinds of questions lead to interesting discussions among scientists. Those who adhere to a purely naturalistic explanation for the existence of life are left struggling to find mechanisms to create such a sophisticated information source. Let's examine what DNA is and how it works to better understand how information is used in the cell. Deoxyribonucleic acid, or DNA, is a long polymer of nucleotides, each nucleotide being a deoxyribose sugar molecule, one phosphate group, and one base. Nucleotides are joined together to form the DNA backbone. A second DNA strand then joins the first to form a double helix. You may recognize the code letters A, C, G, and T, which are the four bases used in DNA. These bases are adenine, A, cytosine, C, guanine, G, and thymine, T. Like the code in Mr. Gates' software, the sequence of the four bases encodes genetic instructions used in the development and functioning of all known living organisms. Once organized, this information is what makes up the gene. And genes, as we all know, contain the recipes for the components of life. Genes hold all the information to build and maintain an organism's cells, and they also pass genetic traits to offspring. Human DNA contains an estimated 20 to 25,000 genes in its approximately 3 billion base pairs. And did you know that if the DNA in an adult human's body were laid out end to end, it would be about 30 billion miles long? That's enough to travel the distance from the Earth to the Sun over 320 times.
The DNA in the cells of my body is the same as that in a fruit fly, an orange, a hippo, an elephant. It's all the same chemically. The uniqueness to all of those species of living things lays in the information content of the DNA. So while the chemistry may be the same, the actual information that's embedded in that chemistry is very, very different. And the challenge for molecular biologists such as myself is understanding completely what that information means.
When a gene is active, the coding sequence is copied in a process called transcription, producing a messenger RNA or mRNA copy of the gene's information. mRNA is produced by a protein that's over 3,000 amino acids long as it reads the genetic code. Wait a minute. How can messenger RNA require a protein that reads the genetic code, but yet the protein needs the messenger RNA to be produced? Could these have both come into existence at the exact same time with the meaningful information needed for both to operate properly? As you can see, much is still a mystery when it comes to the origin of this complex organic language.
And what's fascinating to me, not only do proteins create proteins, but we have proteins that are actually creating the DNA molecule. That's our blueprint. And yet on the DNA molecule, there are sequences, a part of the DNA that are coded for those proteins. So the manufacturing plant needs the blueprint, and the blueprint, the DNA, needs the manufacturing plant in order to actually create the DNA. So what came first, the proteins that made the DNA or the DNA that made the proteins?
Once DNA is organized into a single coiled piece, it's then wrapped around a complex hierarchical structure of proteins. This is what is commonly known as a chromosome. There are 23 pairs of chromosomes in the human genome. A genome is a full set of chromosomes containing all the inheritable traits of an organism. And as we observe our surroundings, we can see there are many different genomes in existence, creating very unique and incredible organisms. Stop to think about our own body having approximately 100 trillion cells, each group having a unique purpose, such as epidermis, the lungs, heart, skeletal structure, muscles, nervous system, digestive system, reproductive system, immune system, and the brain all working simultaneously to form this magnificent machine we call the human body. We begin to have an appreciation for the complexity, elegance, and beauty of life. And as we look around us at the millions of plant and animal species, it becomes incredibly breathtaking to ponder how much information and organization must take place for all of this life to exist.
You know, we're all derived from a single cell, the zygote. And what marvels me is from this single cell, we get 250 distinct cell types, each of which has a very, very unique program. All of the cells, with a couple of exceptions, have a nucleus and have a genome. And that genome is the same across all the cells. So when we begin to think about the complexity of the genome, I think that's amazingly complex, how we go from a single cell to unique cells that have very specific programs of gene expression that are necessary for that unique cell to function. But we're only beginning to understand the information in our genome that directs those unique programs of gene expression.
While we know a great deal about the cell and its functionality, we've only scratched the surface of its inner workings. Scientists are continually discovering new information and data concerning the cell and its DNA. As our knowledge increases, it gives us a deeper understanding of how complex a single cell really is. And as we ponder this miniature marvel, we begin to wonder, could this simple cell have evolved from the elements of the earth over time? Is it possible for this to have happened by natural processes? Possible simply means there is a zero chance of occurrence. However, when a probability becomes too small, it can be deemed operationally impossible or infeasible, meaning that we understand the possibility of it happening is greater than zero, but for all practical purposes, it's recorded as impossible. Now, when something is deemed operationally impossible, it's still technically possible. So some scientists try to justify arguing endless hypothetical scenarios simply on the grounds that they're theoretically possible, and then try to convince the public that something must have happened because it could have happened. An excellent example of this comes from the noted biologist George Wald. He once stated, when asked about the origin of life, "However improbable we regard this event, or any of the steps it involves, given enough time, it will almost certainly happen at least once. Time is the hero of the plot. Given so much time, the impossible becomes possible. The possible becomes probable. The probable becomes virtually certain. One only has to wait. Time itself performs miracles." Let's hope that future generations will develop a keen awareness of this kind of wordplay and use critical thinking to distinguish between probable explanations and pure unscientific speculation.
The fact that you're watching this video shows that your instructor and you value critical thinking and the very definition of science. You want to know. You want to understand the world around you. And it's a beautiful thing to look out there and ask those questions and never allow yourself to be put in a box where you can only think of one possible explanation for how things came to be or how things work. Look outside the box. Develop that critical thinking to look at all the options and say, where does the evidence lead? And what is the best explanation for why this came to be?
So, what is the probability of a simple cell evolving by undirected natural processes? The probability of a single protein being formed by undirected natural processes is only 1 in 10 to the 164th power. That's 10 with 164 zeros following it. That's pretty big. The probability of life, a simple cell evolving by undirected natural processes, is 1 in 10 to the 340 millionth power. That is unimaginable. Here's an illustration to help demonstrate just what this means. Here we have one tiny grain of sand. It's been estimated that there are 1 million grains of sand in a half a cup. It would take 1 million half cups of sand to fill a swimming pool that's 6 feet deep and 30 feet in diameter. Now, if we took 1 billion of these pools of sand, we could fill Lake Tahoe in Nevada, which is about 22 miles long and 12 miles wide. Think you could find that original grain of sand we started with? We're not done yet. It would take about 1 billion Lake Tahoes to fill the volume of the Earth with sand. The probability of grabbing that original grain of sand out of the Earth filled with sand is 1 in 10 to the 30th power. It would take 100 million Earths to fill 1 sun with sand, and 1 trillion suns to fill our solar system, 10 trillion solar systems to fill 1 cubic light year, 100 trillion cubic light years to fill the volume of the Milky Way galaxy, and finally 10 billion Milky Way galaxies to fill the observable universe. Whew. That is a lot of sand. The probability of now randomly picking our original grain of sand from the entire observable universe is 1 in 10 to the 96th power. Still a far cry from our protein being formed at 1 in 10 to the 164th power, and even less chance of life evolving from undirected natural processes at the probability of 1 in 10 to the 340 millionth power. Starting to get the idea? Scientists generally consider anything with a probability of less than 1 part in 10 to the 70th power operationally impossible. And by the way, that calculation doesn't take into consideration the information that's stored in the cell, which directs the cell in all aspects of operation. That estimate is solely the chance of chemicals combining to form something living. So, the chance of life evolving and retaining the information needed to replicate itself is astronomically smaller than anything we know. And that's only one organism. Think about what it would mean for millions of complex organisms to have evolved from an undirected natural process. According to information science, the probability is so small that it's deemed operationally impossible. So, knowing all of this, should the popular scenarios of chemical and biological evolution be taught globally as the only explanation of the origin of life and species? It's my belief that we're not opening our minds to the possibility of other explanations. Now, one of the most basic concepts you should have learned is that if observations and data contradict the theory you're testing, then the theory should be modified or abandoned. Unfortunately, this doesn't seem to be happening to the present, most popular model of origins. Instead, many scientists are trying to take information and make it fit the evolutionary models. But is that good science?
Critical thinking is required to realize the information science aspects of this and to make sure that whatever scenarios you're coming up with do not violate the principles of information science. Too often, scientists, they believe that information can be generated by physical processes, and that simply is not true. Functional information cannot be generated from purely physical properties.
The possibility of life evolving using the known laws of chemistry and physics is operationally impossible. When we consider the laws of information, that possibility now becomes impossible. Meaningful prescriptive information cannot arise from nothing, no matter how much time you allow. And until we acknowledge this, we will never discover the origin of life. Thank you for joining me on this amazing journey through science and the programming of life. I would challenge you to examine the evidences and join the growing community of critical thinkers. Until next time.